Look at your bare foot. If your second toe extends past your big toe, you have been lied to 3 times. The first lie, your second toe is longer. It is not. Your big toe is shorter. The bone behind your big toe, the first metatarsal, stopped growing before the bone behind your second toe. Your second toe looks longer because it starts further forward on a bone that reached further. The second toe did not overgrow. The first bone undergrew. Your foot is not unusual because something is too long. It is unusual because something stopped too early. The second lie, you have Greek ancestry. The internet says this foot shape is called the Greek foot because Greek sculptors carved their gods with it. Having a longer second toe does not make you Greek any more than wearing a toga makes you Roman. The trait appears in every population on Earth. It is not Greek. It is human. The third lie, it means nothing. Ancestry DNA tracked it across 930 genetic markers. 930. That is nearly as genetically complex as human height. Your body did not invest 930 markers in a trait that does not matter. Evolution does not maintain complexity for nothing. Something about your second toe mattered enough for 930 points in your genome to keep building it. Subscribe. Because the trait on your foot has been misnamed, misattributed, and misunderstood for 2,500 years. The DNA just set the record straight. The medical name is Morton's toe after Dudley Joy Morton, the American orthopedic surgeon who first described it in the 1920s. The clinical mechanism is brachymetatarsia of the first ray, which means premature closure of the growth plate in the first metatarsal bone. Your foot has 5 metatarsal bones, 1 behind each toe. During development, each bone grows at its own rate until the growth plate closes and the bone reaches its final length. In most people, the first metatarsal, the one behind the big toe, grows to be the longest or second longest metatarsal. In roughly 10 to 30% of the population, it does not. The growth plate closes early. The bone stops, the second metatarsal keeps growing. And the result is a big toe that sits further back than the second toe. This is not damage. It is not a defect. It is a genetically programmed timing event. The growth plate received the signal to close before it finished growing. That signal was sent by your DNA. And the timing of that signal varies between individuals because the genetic variants controlling it are distributed unevenly across populations. Ancestry DNA can predict whether your second toe is likely longer than your first from a saliva sample, not by looking at your foot. By reading your genome, the 930 markers they identified are scattered across multiple chromosomes, each contributing a small effect to the timing of first metatarsal growth plate closure. If you inherited enough of the early closure variants from both parents, your growth plate stopped early. If you did not, it kept growing. Only 13% of the variation in second toe length is currently explained by the identified genetic markers. That means the other 87% involves genes that have not yet been found, environmental factors during development and interactions between genes that are too complex for current analysis to resolve. The 930 markers are the beginning. The full genetic architecture is larger. Your foot is carrying a trait that is genetically as complex as height, only partially mapped and present in every population on Earth. The growth plate that quit early was following instructions. The instructions are written across 930 known markers, and the full instruction set is still being decoded. The ancient Greeks considered the longer second toe the ideal foot. Not one ideal among several. The ideal. The divine proportion. The shape that sculptors were trained to reproduce when carving gods, goddesses, and heroes. The Venus de Milo has it. The statue of Zeus at Olympia had it. The caryatids of the Erechtheion have it. Michelangelo's David, carved 2,000 years after the Greeks but using their proportional standards, has it. The Statue of Liberty, designed in the 1870s using classical Greek proportions, has it. The most famous foot in America was designed with Morton's toe. For 2,500 years, from Athens to Rome to Florence to New York Harbor, the longer second toe has been carved into humanity's most important sculptures. The sculptors were not depicting a medical condition. They were depicting perfection, and their definition of perfection required that specific toe to extend past the first. The question nobody asked for 2,500 years was why. Why would Greek sculptors, independently from Roman sculptors, independently from Renaissance artists, independently from 19th century French designers, all arrive at the same conclusion about what the perfect foot looked like? If you have made it this far, subscribe, because the answer is not about aesthetics. It is about proportion. The longer second toe creates a more tapered foot profile. The foot narrows gradually from the arch to the tip, rather than having the blunt, squared-off appearance of the Egyptian foot, where the big toe is longest. In visual art and proportional theory, the tapered form is considered more dynamic, more directional, more suggestive of motion. The Greek foot looks like it is moving forward even when it is standing still. The sculptors were not depicting a trait they had, they were depicting a proportion they found beautiful. And the proportion they found beautiful, the tapered, forward-pointing foot, happened to correspond to a genetic trait that 20 to 30% of the population carries. The art did not create the trait existed, the art noticed it, and for 2,500 years, the art elevated a growth plate timing variation into the foot of the gods. Your big toe is the most important structural component of the walking foot. During normal gait, 40 to 60% of your body weight passes through the first metatarsal and big toe during the push-off phase of each step. The big toe is the launch pad. Every stride you take is propelled by the bone that, in your foot, stopped growing early. When the first metatarsal is shorter than the second, the weight distribution shifts. Instead of loading primarily through the first ray, the force transfers to the second metatarsal head, the ball of the foot behind the second toe. The second metatarsal is thinner. It is not designed for primary load bearing. The consequences are measurable. People with Morton's toe have higher rates of callus formation under the second metatarsal head, higher rates of metatarsalgia, chronic pain in the ball of the foot, higher rates of second metatarsal stress fractures, and in some studies altered balance and gait efficiency on flat surfaces. But here is where the narrative gets complicated. A 2026 clinical trial, registered at clinicaltrials.gov, is currently studying whether Morton's toe morphology is associated with superior neuromuscular performance in female football players. The hypothesis is that a longer second metatarsal may provide a wider base of support during lateral movements, potentially enhancing agility and balance during rapid directional changes. The same trait that increases stress fracture risk on flat ground may provide an advantage on uneven terrain or during lateral sport movements. The foot that is less efficient for straight-line walking may be more stable for cutting, pivoting, and changing direction. This dual nature, disadvantage in one context and advantage in another, may explain why evolution maintained the trait at 20 to 30% rather than eliminating it. If Morton's toe were purely disadvantageous, natural selection would have reduced its frequency over the 200,000 years of modern human existence. If it were purely advantageous, it would be universal. Instead, it sits at a frequency that suggests it is beneficial in some environments and costly in others. A balanced polymorphism, a trait maintained by competing pressures. The flat ground says it is a problem. The uneven terrain says it is a solution. And 930 genetic markers say evolution could not decide. The frequency of Morton's toe varies dramatically across populations, and the variation is not random. The highest documented frequency belongs to the Ainu people of northern Japan. Roughly 90% of Ainu individuals have a second toe longer than the first, 9 out of 10. The Ainu are descended from the Jurmen, the prehistoric inhabitants of the Japanese archipelago who arrived roughly 14,000 to 30,000 years ago and represent one of the oldest continuous lineages in East Asia. The Idoma people of Nigeria show a prevalence of approximately 37%, based on a study published in the Scholars Journal of Applied Medical Sciences. European populations average 20 to 25%. East Asian populations outside the Ainu tend to show lower rates. If your parents both have Morton's toe, your probability of having it is roughly 60 to 70%. The familial inheritance is strong but not absolute, consistent with a polygenic trait where multiple genes contribute and the environment plays a role. The population distribution follows a pattern that correlates with ancestral terrain rather than ethnicity. The Ainu lived on volcanic islands with steep, rocky, uneven terrain for thousands of years. A foot with a shorter first metatarsal distributes weight across a wider forefoot platform, which may provide better stability on slopes and irregular surfaces. Populations that spent millennia on flat savanna maintained longer first metatarsals optimized for efficient straight-line locomotion. The terrain shaped the foot shaped the gene pool, and the gene pool carried the trait forward in proportions that reflect how flat or how rugged the ground was under your ancestors' feet. Your second toe is not longer because of grease, it is longer because of terrain. Terrain your ancestors walked on thousands of years ago in a landscape you may never have visited, on ground that selected for a bone structure you carry without knowing why. So look at your foot one more time. If your second toe extends past your big toe, here is what the science says you are carrying. A growth plate that closed early in your first metatarsal, triggered by a combination of variants scattered across 930 genetic markers in your genome. Only 13% of the variation has been explained so far. The remaining 87% involves genes not yet identified and developmental factors not yet understood. The full genetic architecture is larger than what has been mapped. Your foot is carrying instructions that science is still learning to read, a trait that has been idealized in Western art for 2,500 years. carved into the Venus de Milo, Michelangelo's David, and the Statue of Liberty. Not because the sculptors had the trait, because they considered the tapered proportion more beautiful than the alternative. The art noticed the trait. It did not create it. A biomechanical profile that disadvantages you on flat ground but may advantage you on uneven terrain and during lateral movements. A trait maintained at 20 to 30% across the species because evolution could not determine whether it was a problem or a solution. The answer depends on the ground. A population map that peaks at 90% in the Ainu of Japan and averages 20 to 25% in European populations. The frequency reflects ancestral terrain, not ethnicity. Rocky volcanic islands produce high frequencies, flat savannas produce low ones, and a genetic complexity that rivals human height. 930 markers, polygenic inheritance, no single gene responsible. A trait so genetically expensive to maintain that evolution invested nearly as much code in your toe length as it invested in how tall you are. The Greeks looked at this foot and saw divinity. The orthopedic surgeon who named it saw a condition. The geneticists who mapped it saw 930 markers. And the foot itself has been carrying all three of these interpretations, plus the terrain map and the growth plate timer and the biomechanical trade-off for your entire life. Every step you take, you walk on a trait that is simultaneously a work of art, a medical category, a genetic puzzle, and a fossil record of the landscape your ancestors navigated, all in one toe, that is not longer, on a bone that stopped too early, 930 genetic markers, a growth plate that quit early, a trait carved into the gods by Greek sculptors for 2,500 years, a biomechanical trade-off between flat ground and uneven terrain, 90% prevalence in the Ainu, 20 to 25% in Europeans, and a genetic complexity that science has only 13% decoded. Your second toe is not longer. Your big toe is shorter. And the reason it is shorter is written across 930 points in your genome that your ancestors carried through terrain you have never walked on, past sculptures you may never have seen, and into a body that has been carrying the answer on its foot since the day it was born. Drop a comment. Is your second toe longer? Does it run in your family? And after this video, does the foot you have been walking on your entire life feel different than it did 16 minutes ago? Subscribe for more, share this with someone and make them look at their feet. We will see you next time.